You ever wonder how scientists can test a brand new drug or figure out how to make a vaccine without using a person for every single tiny experiment? Well, the answer is kind of mind-blowing. They've learned how to grow living cells outside of the body. Today, we're going to go on this incredible journey, following a cell from its home inside a living thing to a whole new life in a lab.
Right, so this is the central question, isn't it? It's what drives so much of modern medicine. If you can't actually see what's happening inside a living, breathing system, how in the world do you figure it out? How do you test your ideas? The solution is a cornerstone of biology, and it all comes down to creating a whole new world for life to thrive in. And there it is, the answer. Scientists literally build a miniature, artificial world for cells inside a petri dish. This whole amazing process is called cell culture, and honestly, it's the key to just about everything we're going to talk about. So let's start our journey. Let's follow a cell as it makes its way from living tissue to a brand new life in the lab.
So, our journey kicks off with what I like to call the Great Cellular Escape. This is the first and maybe the most dramatic step. Before you can study a cell, you have to get it out of its home, that super complex neighborhood called tissue, and somehow convince it to live all on its own.
Okay, so what you're seeing here is basically the roadmap for this whole escape plan. It all starts with a little piece of an organ. From there, the cells get isolated, then they're encouraged to grow in their first new home, and eventually, some of them can even go on to form a stable, long-term population. This is our guide for the journey ahead.
So the very first challenge is that cells in a tissue aren't just hanging out. No, they're all stuck together with this kind of biological superglue. In the process of breaking them apart, that's called disaggregation. Just think of it like trying to gently persuade a really, really tightly knit community to go their separate ways.
And scientists have two main ways of doing this. On one hand, you've got the mechanical method. This is kind of the brute force approach. We're talking chopping, slicing, physically breaking the tissue apart. It's fast, for sure, but it's rough, and you risk damaging a lot of your cells. Then you've got the more, elegant enzymatic method. This uses special proteins, these enzymes, to just gently dissolve that biological glue. It's slower, yeah, it's more delicate, but the reward is often a much healthier population of cells to work with.
Now, the undisputed king of these enzymes is something called trypsin. The best way to think about it is like a pair of microscopic molecular scissors. It just goes in and snips all the little protein connections that are holding the cells together. It's incredibly effective, and a whole bunch of different cell types can handle it.
But even when you decide to use trypsin, you've still got choices to make. You can use a warm method, which is super fast and intense because the enzyme is really active at body temperature. Or you can go with a cold method, which is a much slower, more patient approach. You see, the temperature here makes all the difference. It controls the speed and intensity of those little molecular scissors.
This table here just breaks down those trade-offs perfectly. The warm method is quick, right? Done in a few hours, but that intensity can really stress out the cells, which means fewer of them might survive. The cold method, on the other hand, is a marathon, not a sprint. You do a long cold soak that lets the trypsin seep in gently, and then you do a short burst of heat to activate it. It takes way longer, but the payoff is a much higher yield of happy, healthy cells. It's that classic choice you see in science all the time, speed versus quality.
Okay, so our cells have done it. They've escaped. They are free from their tissue. But now, they face a new, and honestly, even bigger challenge: surviving in a completely alien world. This first generation of cells growing in a dish is what we call the primary culture.
And this is a really crucial first step in their new life, the primary culture. The key thing to get here is that these cells are fresh out of the body. They've never been grown in a lab before, and that makes them incredibly valuable because they're an almost perfect reflection of the tissue they came from. So, to survive, these cells need a very specific recipe. First, most of them are what we call anchorage dependent, which just means they need a surface to stick to, like the bottom of a plastic flask. Next, they need a controlled environment, an incubator, that basically mimics the warm, cozy conditions inside the body. And most importantly, they need food, this liquid called culture medium, which is like a perfectly balanced, custom-made buffet of every nutrient a cell could ever want. And here's why primary cultures are so precious. At this early stage, the cells are essentially a perfect genetic snapshot of where they came from. They have the same number and look of chromosomes, what scientists call a karyotype. This means they behave almost exactly like they would inside the body, which for research is absolutely fantastic.
But this perfect, ideal state, it doesn't last forever. The primary culture pretty soon reaches a critical turning point, a cellular crossroads, and the cells have to choose one of two very, very different destinies.
See, as the cells grow and multiply, they start to run out of space. When the dish gets too crowded, scientists have to split the population and move some of them to a new dish. That process is called subculturing. And the moment you successfully do that, you've crossed a major threshold. It's not a primary culture anymore. It has now officially become a cell line. And this is where those two destinies really come into play. On the left, you've got finite cell lines. Just like most normal cells, they have a built-in expiration date. They can only divide a certain number of times, maybe 20, maybe 100, before they just get old and die. But on the right, you have continuous cell lines. These are the rock stars of the cell world. They are, for all intents and purposes, immortal. Either through some genetic tweak or because they came from a tumor, they've gained the ability to divide forever.
So, we followed our cell on this wild ride from tissue to potential immortality. But why does any of this matter? What's the big deal? Well, it's because these cell lines aren't just a lab curiosity. They are the invisible engines driving a huge portion of modern biology and medicine.
Let's meet some of these cellular celebrities. You've got Vero cells, which come from monkey kidneys, and they are absolutely crucial for making safe vaccines for diseases like polio. There are HEC293 cells, a total workhorse for countless research labs studying basic human biology. And then you have CHO cells from Chinese hamster ovaries, believe it or not, and these are tiny biological factories that produce many of the complex life-saving drugs that treat things like cancer and autoimmune diseases. These cells are literally saving lives every single day.
And that brings us to our final thought. We figured out how to grant cells immortality in a plastic dish. So the question we're left with is a pretty big one. By studying these cells that can live forever, what secrets might we unlock about aging, about disease, and maybe, just maybe, about longevity in our own bodies? The journey of the cell, it seems, is far from over.